Hi, and thank you for joining us on an episode of Learning with Caleb. My name is Caleb Andrew. I'm a student in the human embryology class with Brother Bailey, and there's no doubt that you have already watched my partner's presentation on the development of the eyes. I will be talking about the development of the ears throughout pretty much weeks 4 through the 9th month of gestation. So, what I would like to do is split up the ear into its 3 major components, and I'm going to write these down and show them to you. So what we're going to do, like I said, first actually, we're going to talk about its start and finish. Starts usually around the 4th week and ends usually around the 9th month, as you can see right there. Now, let's split it up into its 3 main parts. We have first the inner, the middle, and the external ear. These three main parts are what comprise our ear, or at least the ears that we have now. And I will talk in detail about how each one of these forms. So I'd like to first begin with the inner ear. The inner ear is what allows us to have balance. So let's write that. So the inner ear is what gives us balance, and it is what converts chemical signals into electrical signals, which are then sent to our brain. We'll talk about how how the different parts of the inner ear are formed, and how they contribute to those electrical signals that are then sent to our brain. Next is the middle ear. There are a few components I wish to discuss with this, actually really just 2. We first talk about the ossicles, and then we talk about the tympanic cavity. As you can see here. The ossicles are the bones that are connected to your eardrum. Those bones connected to your eardrum are what connect the external ear to the inner ear. Those ossicles are called the stapes, the incus, and the malleus. The tympanic cavity is the cavity that surrounds those ossicles, keeps them in place, and forms during this 4th week to the 9th month of gestation. Lastly, we would like to talk about the external ear. The external ear comprises of the meatal recess and the auditory auricle. So as you can see here, we have the 3 different components of the ear. And I'd like to talk about each one. I'd like to begin with the inner ear. So, talking about the inner ear is primarily, actually, I think entirely, ectoderm derived. And what we have here to begin with is we have the formation of what is called our otic pit in the ectoderm. This is caused by the invagination of the ectoderm into the embryo. This is induced by FGF signaling. So this is called our otic pit. And kind of the cell lining around it is called our otic disc or otic placode. So what's going to happen is as FGF signaling induces the invagination of this pit into the embryo, it's going to start to enclose around it until it pinches it off. We get programmed cell death, and those cells will then enclose the otic vesicle, and we will get the primary vesicle, which will then elongate, grow, and turn into our inner ear. So we're going to continue on with learning about this and the different parts of the otic vesicle and all this different stuff. But first I'd like to talk about how we divide up the otic vesicle. So we divide it up into its dorsal and ventral surfaces. So I'm just going to write D and V. And with our ventral surface, really quick, we actually get what is formed our cochlea and our saccule. With our dorsal surface of the otic, or dorsal part of the otic vesicle, we get the remaining structures. As you can see here. These are just the cochlea and saccule are just the inferior part of the inner ear, and the remaining are all the superior parts of this. So as time goes on, we get the elongation of the otic vesicle. Kind of looks like that, just a little strange looking. This part up here, so these three parts have certain names, which we will identify here. We have the endolymphatic sac, pars superior, and pars inferior, as shown here. These will continue to grow and to fold and to turn into different structures. And we will ultimately get a structure that looks kind of like, to be honest, a convoluted pretzel. Like this. Now I can identify the parts and all that and everything that's going on. We have the cochlea down here, the ductus reunions, the saccule. We have our endolymphatic sac, which is right here. This part becomes this, and then we get our posterior, or our anterior, and our lateral semicircular canals. And these are all created by the otic vesicle, and this is all ectoderm derived. So we will then go into the different parts of this. So now I want to go over something really quick, just kind of the way we hear and how and what cells develop. So what happens is we will get, I'm going to use a different color here. We will get in the cochlear duct, there's a window, or the cochlea, there's a window from which the stapes is actually connected to. So you have the stapes here. I'm just going to explain this really quick. And then this is our cochlea. So here's our stapes, there's the incus, here's our window that it is attached to. So as sound waves hit the eardrum, those move these ossicles. The ossicles then move this window. The cochlea has fluid inside, and as the window is moved, it causes waves to move throughout the cochlea. Those waves are picked up by a certain kind of cell. So I'm going to those waves cause movement, and those movement actually open up channels for which potassium can enter into a cell. That potassium then converts into neurotransmitters, which then are sent to axons, and then those electrical signals are then sent to the brain. I'm just going to draw a quick picture of those hair cells. Those hair cells just kind of look like that. They kind of look like these progressing steps. And there is a surface, so there's a surface right here. As it moves those cells and opening up those channels, and thus allowing those ions and everything to move into the cell, turning the chemical into electrical signals. I'd like to talk about something that can apply a little bit here. There are people out in the world that have hearing loss. There's a lot of people that have hearing loss. Actually, I believe it's every 1 in 1,000 births have some sort of malformation that produce hearing loss. One of the types of hearing loss that has to do with the inner ear has a certain name. So it is called sensory neural hearing loss. That's what the HL stands for. So sensory neural hearing loss has to do with the inner ear. Now, usually, an example of sensory neural hearing loss usually has to do with the vestibulocochlear nerve. Now, that nerve is responsible for taking those signals and sending them to the brain. And a type of hearing loss that has to do with sensory neural is called vestibulocochlear dysplasias, in which you are missing part or all of your inner ear. Now you can imagine what kind of problems that could create. Loss of balance, really couldn't hear at all unless somehow we could create a synthetic version of the inner ear that we could then place inside someone's head, which I haven't done the research on that, but I don't think we have anything for the moment. So this is a very serious type of hearing loss. And so you get that loss of balance and everything like that. Now, with that, just like to talk about a few things with the sensory neural hearing loss. So I'm just going to call it SNHL. With that, you get a deficit, pretty much, of the transfer of chemical to electrical. The chemical to electrical signaling. You get a lot of, because of that, nothing's registered in the brain, and you also get a loss of balance, as I've explained before, and you can get these dysplasias, which you are missing part or whole of the inner ear. Another important part of the ear, and this is where I'm going to change, I'm going to shift into the next part of my presentation, actually has to do with the middle ear. Now the middle ear